The speaker. Uh, okay, so good morning, ladies and gentlemen. It's a great pleasure for me to introduce our speaker for the first session of the day, Professor Ananthasuresh, who is going to talk to us about a very interesting topic, grasping cells. The talk itself will be definitely of interest to both the biologists and the engineers alike. Our Professor Anand Suresh, he earned his PhD from University of Michigan. So before his PhD, he did his master's from University of Toledo and bachelor's degree from Indian Institute of Technology, Madras. His principal research interests are compliant mechanism and topology optimization and applications in the micro electromechanical systems. Very interestingly, he also works on the biomechanics of the cells, biomedical devices, and micro robotics. So in addition to research output in terms of more than 275 journals and conference publications and 16 patents, his research group has also spun off 4 startups, such as Pat showed. He currently serves as the professor of mechanical engineering and the dean of the division of mechanical science at Indian Institute of Science, Bangalore. So previously he was the chairman of Center for Biosystems Science and Engineering. He has received many awards, among which including the prestigious Shanti Swarup Bhatnagar Award way back in 2010 for the engineering science. So definitely this is a kind of a fanboy moment for me to welcome Professor Anand Suresh. So when I was doing my PhD with MBO at IISc Bangalore, we used to look at Professor Anand Suresh lab and his research with great awe. Because he was the one who kind of introduced this interdisciplinary approach in India, I can say actually. And we used to visit his lab to look at the different experiments he used to carry on with the biological system. So, ladies and gentlemen, please join me in welcoming Professor Anand Suresh. Sir, now the stage is all yours. So, thank you once again.
Yeah. Okay. Thank you, Tageshwar, for the kind introduction. I didn't know that you were in MBU here. Um, yeah, very good to see all of you, or at least online. And Tageshwar, how long is this going to be?
Sir,
How much for how
we
much? Yeah.
have time till 12, sir, actually. So you can give a talk till 12:30 or something.
Okay. Okay. Sure. Sure. Yeah. And I guess it's a small group, and you can interrupt me during the talk, so that I turned off my video, but anyway, you know that I'm here. You can interrupt and ask questions in between also, so that we'll make sure that uh, we can interact here rather than just in a one-way talk, for which you could have seen my videos also. Anyway, thanks to NIT Warangal for inviting me for giving this talk. This is something that we've been doing for quite some time, and it is changing over the years. So I just call it grasping cells, and I'll tell you, why we call it grasping in that sense. So this work was done with a number of PhD students and project staff. Of course, my colleague Prof. Samitra Das in microbial cell biology in IISc is the kind of provider of the biology side of things, and my one of the first PhD students, Professor Narayan Reddy, who was at Gauhati until a couple of years ago when he unfortunately passed away. He started this computational work, and I had a master student, Deepak Sahu, who is now working for General Electric. He was one of the first people to do a miniature gripper to grasp a cell. And Santosh Bhargav is a PhD student. Now he founded a company called Benflex, and then another company called Spuvum. That's the combination of the word sperm and ovum, where he uses these biomechanics and miniature grasping tools for this application that he does this fertility chip. And Srinath Balakrishnan was one of the recent PhD students who is now an assistant professor in IIT Goa, and he has been doing this, continuing this biomechanics work. And I'll be describing some of the work that these students have done. Also, I had another project assistant, Somana Kolimada, and then I have current students and postdocs, current Anvesha Barua, postdoc Shilpa Ramachandran, and Sudhanshu Shekhar, and Wageshing, another PhD student, Prasenjit is one more PhD student. So you see, except Anvesha and Srinath, who are PhD students in our biosystem science and engineering, rest of them are actually students in mechanical engineering. So you can see how we can excite mechanical engineering students to think about biological cells. But of course, whatever we do is still mechanics and mechanical engineering. It's just applied to biological cells. So I asked this question. So if you were to hold a single biological cell, like I've shown, of course, with your hand of this size, I hope you can see my cursor. If you have a hand of this size, cell will not be of this size. Cell is of micron dimension. So you need to make a hand that is of micron sizes if you want to really hold. Okay, let's say we do that. We can actually do uh, tools that with which we can hold a biological cell. I'll describe that now. But the question to ask is, what can you learn if you can literally grasp a cell? So you can hold a cell like you would hold an onion or a tomato or whatever, right? What can you say? One thing that we can actually say is that if you go to buy tomatoes, you would try to feel them, right? How does it feel? Does it feel soft? You wouldn't buy, right? Because it will go bad very soon. It's very ripe. On the other hand, if it is too hard, also you wouldn't buy because you have to wait for a week or so for it to ripe. So you want to get the right kind of tomato. You just feel it, right? So what I'm saying is that if you were to hold something and try to characterize its mechanical response, in this case, stiffness or hardness, stiffness or flexibility, or hardness or softness, you can infer by holding it. And when you hold it also, if you just imagine how you buy tomatoes, right? You don't hold it one way and then leave it. You actually hold it this way, that way, and then you try to manipulate that and then see how it feels overall. Okay, that's what we naturally do these things. So we try to do that to the biological cells. If we do that, what we can say is that whether it is normal or not normal. Just like we say, a normal tomato is not too soft, not too stiff. Okay? So that is what we can sense. In a similar way, if we know what a normal cell feels like and what an abnormal cell, or abnormal in a sense, it has some kind of a disease state in it. Okay, so can we tell that? And that's what we are saying. So what can you learn? We can learn whether it is normal or not normal. Okay, but we don't want to stop there. We can do more. Now let's say instead of buying a tomato, right, by feeling it, uh, now let us see what a doctor does. Okay, when you go to a doctor, when someone goes to a doctor with some abdominal pain or whatever, right, doctor would feel it. Doctor would feel the abdomen to see if everything is all right. Okay, so they have a sense of knowing what is normal, what is not normal. Okay, so can we do that? But then when we started doing it, biologists, the first thing they say is that, oh, biology is very heterogeneous. Okay, you have to have specificity. That is, if something feels soft, it can be soft for many reasons. A tomato, when it is soft, we can say it is too ripe. Okay, that's all there is. Okay, but cell is not like that. There are many factors that actually decide the stiffness of a biological cell. And we don't know a lot of causes also. If it becomes stiffer or more flexible than usual, then there'll be many factors. You have to specifically say that this is what did it. Okay, and that's where we kind of started our work. How do you establish that specificity in the stiffness of biological cells so we can actually understand? And that's why the first page is 20 the computational work, which I'll be describing a little later in the talk. Yeah. So if you see, a cell will have a cell membrane and there'll be lots of things. A cell is really crowded, you know, all this. Okay, things will be floating around. Okay, So a cell is a very complicated thing. I would actually call it granular matter, in the sense that there are a lot of particles inside the cell which are floating around in the cytoplasm, which is a liquid. Okay, and there are strings and all these. When I say strings, there is microtubules and actin filaments and intermediate filaments and so forth. Basically, there'll be lots of things. So Francis Crick, who actually tried to do it. Okay, so he actually described a cell as a mother's work basket. Okay, it is a jumble of beads and buttons and all kinds of pins and threads, and they're all jostling about, okay, held together by some forces. That's what Francis Crick described it in his paper written in 1950. 1950 is an important year, because just 3 years later, uh, Crick and Watson discovered the double helix structure of DNA. Okay, so while he was working on such a fundamental thing, he was also looking at other things just a few years before. Okay, so busy people are busy with many things. There are a lot of things that they do it. He had written a two-part paper, which is nice, one experimental and then theory. Okay, but it's interesting. This is volume one, issue one. Okay, experimental cell research. Right. So he compared this work back. That's important to remember. There are all these things inside. It's a very complicated thing. If any of those, okay, let's say this pin or a thread or a button or a bead, okay, those are of course cell organelle. There's mitochondria and this and that. Lots of things are there in the cell, right? So if any of those things change, let's say mitochondria, for example, is broken into little pieces instead of being one big thing, wouldn't the stiffness change? Okay, can we specifically say that that is the reason? All right, so those are the questions that we want to ask now. Anyway, that's how the Crick's view was of a cell, which is a correct way. In fact, now one of my PhD students is looking at modeling a cell as a granular medium using a technique called discrete element method. Discrete element method is developed for modeling granular medium, like such as sand, for example, a grain of rice. Okay, so grain of rice, like liquid, it'll take the shape of the vessel, because all the little particles, the grains of rice will go, right, or sand, and it also flows, right? So sand would flow, and there are instabilities, interesting dynamics. That's how we are actually looking at it. But coming back to Crick, so this is the excerpt from the Crick's paper. So he's talking about twisting a cell, dragging a cell, and prodding a cell. Okay, so he actually thought about doing these interventions on a cell so that its mechanical characterization can be done. Okay, this is the paper. Again, I'm just showing that physical properties of cytoplasm is what he did with what he called magnetic particle method. Today, twisting magnetocytometry is what people call it. Okay, they apply magnetic particles and do it. So he was a forerunner of that technique in 1949 and 50. Okay, and this is actually the quotation that I just showed you, how he described the cell. He said, if I were to compile just a model, that's how he would describe it. Okay, now any of those things change, stiffness changes, we can actually see that it is not normal. That's where we are starting. And if you think 1950 was way too early for this to do, there were earlier papers. There were 1926 and 1939, 1931. Different people have looked at it, looking at the physical property, they call it. Today we call it mechanical, more specific, not just general physics, but mechanical. And if you actually think about it, it goes back to Robert Hooke's time. That is mechanics and cell being together. So Robert Hooke is the one who gave us this funny acronym, uh, or abbreviation, that he not acronym, really like a simple adage, would tensiosi quiz, which I put an anagram, actually put all these letters in alphabetical order and gave this. He was very secretive. Okay, so he put this thing as an anagram, okay, which actually means that force varies as a stretch, which is a fundamental law in material modeling mechanics, which we call Hooke's law. The same Robert Hooke is the one who looked at Cork cells and coined the term cell, saying that biological matter is made of building blocks, which are cells. Okay, just like atoms are the building blocks for all inanimate matter, which is a fundamental discovery. So both the fundamental law of inner elasticity and the cell biology go to the same person, Robert Hooke. So cell biology and these okay, anyway, this blocks a little bit. I don't know how to get it. Okay, it went away. Uh, so cell biology and mechanics of solids were coupled with each other when both were born in a way. Okay, for both of those, uh, Robert Hooke was the father. Okay, in a way. So this is not really something. Now, let me come to what I'm going to describe. The first thing I want to talk about is what we do when we couple mechanics and cell biology. Not so much of cell biology. After all, I'm a mechanical engineer. My students are mostly mechanical. We try to look at biological cells as mechanical entities, which is not wrong actually, okay? Because in some sense, biology cannot escape physics and hence mechanics. So anything inside a cell changes, it is likely to bring in changes in the way the cell would respond to mechanical stimuli or even biological stimuli. Okay? So if you were to hold a cell and move it, it feels different if it is not normal. In a similar way, there could be biological or biochemical cues which also can change the mechanical response of the cells. Okay. In fact, my former student Srinath Balakrishnan, who is now a professor in IIT Goa, he always finds it interesting that MECH, MECH, and CHEM, chem, are very close to each other. Both relate to biology. No? Now we have biochemistry and chemical biology. Now we have biomechanics and mechanobiology. So chem and mech. So lots of things that happen in biology can be interpreted in terms of chemistry, but also physics or mechanics. Okay? So now we want to actually explore. We've been thinking about doing this mechanodiagnostics. Can we actually say that something is not right, something is not normal, something there is a disease condition? Okay, can we do that using mechanical response rather than biochemical response? Now, whenever we have any diagnostics, right? So in fact, Path showed that Yogeshwar said, actually, that Path is my colleague's company. My students have started this Bentflex and Spoem and other things. Okay, so it is not uh, biochemical diagnostics that we want to do. We want to do mechanodiagnostics. Okay, and the process we also can study what people call today mechanobiology, studying the effects of forces on cells. Okay, and our idea is to understand the underlying processes. Okay, using computational approaches, uh, modeling some inverse problems like we do for our engineered systems or inanimate things. And other is mechanical intervention. Mechanical intervention, just like we have interventions that doctors would do when you go to a hospital, right, for diagnosis as well as for treatment. Similarly, can we do these mechanical interventions to biological cells? Grasping is definitely an intervention. Okay, so just like under a hood of a car, you would try to look at diagnostics. Something is wrong or right, right? So we want to do the same thing to biological cells. And for that, we need these tools. So we cannot use a spanner or a wrench, right? So to do this, we need to have some other set of tools, and we are developing those tools for quite some time. Ultimately, what we'd like to do is biomechanical assay. And we think it's a good alternative to biochemical assay for a few reasons. One is that it's going to be much less expensive. Whenever you go for a diagnostics test, you pay considerable amount of money. In fact, that's one of the worries that we have when you go to a hospital, that will be overdiagnosed. All kinds of tests are done. It's inconvenient and also it's expensive. But now we are not going to use any chemicals. We actually pay for the chemicals when you pay partly for the chemicals whenever you pay for a blood test or any other test, other than imaging, of course. Okay, so here we don't need chemicals. We're just feeling and doing it. Of course, we have a lot of work to do, but our vision is that we can do it with much less cost. Okay. Second, the sample size could be much smaller. We're not talking about a drop of blood. We're talking about a few cells. Okay. It can be much smaller than a drop of blood. And it'll be quick. It just feels. No? How long does a doctor do abdomen examination? Or if you go with a swollen arm, doctor would just hold it and then know immediately. In a similar way, it'll be very quick when you do mechanical characterization. It'll be portable because it's going to be small devices. I'm going to show. Okay, there are reasons why mechanical assays might be useful in the future. Of course, a lot of work needs to be done to get there. So what I'm going to describe now is how do we measure this mechanical response of cells? What kind of tools that we are making? And second is what do we make out of the mechanical response? Okay, we measure the force and displacement and so forth. What can you make out of that? Okay, some computational techniques are needed to make some sense out of those. And then can we actually figure out some biological implications of that? That's what I'm saying. If anything change in the cell because of biological reasons, that would change the response, right? If we have a rotten tomato, what has happened inside is actually biological change. Okay, it is rotten. Now we can actually feel it that it is becoming overly soft, right? So that's what we want to do. Biological implications of change in mechanical response, and of course the tools to do these interventions. Okay, that's what I'll describe today. So our tools are these manipulation tools for cells where we can actually hold a cell. I'm showing my hand, but actually it's a version with which we can hold it, rotate it, squeeze it, stretch it, all kinds of things, okay, that you can think of. And also have these sensors to measure these things. Okay, they're all mechanical sensors, as you will see. We don't use chemicals, no, nothing biological, chemical, everything is mechanical. Okay, because what we are sensing is force, strain, stiffness. They're all mechanical parameters. And other thing is when you are doing this, we also started thinking about this in vivo conditions. That is, whenever you test something outside, which is how most of the time biology experiments are done, they are basically called in vitro in glass, petri dishes. Now, of course, plastic. Okay, so these are all done in outside the living environment. But then cells that are inside the body are feeling an environment which is called extracellular matrix. A lot of mechanical cues now people are figuring out the cells sense and they react to it. So we want to do such things. We have developed some tools where we can imitate these in vivo conditions because inside, um, for example, if you take heart cells or lung cells, they are always under kind of motion. Okay. If you take liver cells, there is perfusion. Whereas if you do in a static culture dish, where you're taking a dish and putting some cells, putting a medium that is static. So the medium is static, it's not flowing, and we have substrate on which we grow the cells is static. That's not true inside. Of course, it's mostly 2D when you do it in the lab. Now we have to go to 3D. So those are the things that we're trying to build the tools so that we can create those in vivo conditions. Okay? Then there are techniques that we use. We do, how do you design these tools? Or how do you fabricate them? It's microfabrication because they're small, right? If I want to hold a cell that is of micron size, my hand should be of micron size also. So we design such tools. And then we do this mechanics, in terms of inverse problems. If you measure this, what is the implication of something else? Okay, if you doctor press on the abdomen, doctor is actually feeling the force and display with the palm, and then try to see whether it is right or wrong. You have to do some inverse problems. What you do in your mind when you're buying tomato, you have to do it mechanically. Okay, and the continuum modeling using finite element method, and also discrete modeling using discrete element method. These are the things that we have used: computation and some engineering. And I'm going to describe our work with two case studies. One of them is old, another is kind of ongoing. So with a specific thing, the one is related to breast cancer, and more like treatment, therapeutics. Other is fundamental, in terms of understanding the biological implications of hepatitis C virus on liver cells. Okay, first our tools. So our tools are called compliant mechanisms. Okay, so I don't know the background of the audience. If you are a mechanical engineer, you might be able to appreciate it better. That normally mechanical devices that we use would have rigid bodies and joints, whereas we do not have anything that has joints. The whole thing is one flexible continuum, which we call a compliant mechanism, and that will behave like a machine or a mechanism. So there are lots of things, including proteins, for example. So uh, Professor Tageshwar mentioned MBU. MBU has a lot of people working on proteins. I collaborated with Professor Ananthasuresh and MBU looking at protein design as well. Proteins for me are nanoscale compliant mechanisms. They don't have joints, they're flexible. And of course, here is the SU8 polymer based microgripper. And these are the cells. I'm going to show videos of these. Of course, we can make with silicon also. We can make with metal. And this is the force sensor that we have developed. So all these compliant mechanisms at macro scale also we work on all the way to nanoscale. So all these tools become useful for our study. And how does it work? This jerky video is showing that when you are pushing over here, how this will come together and then grab something. And there are no joints. The whole thing is flexible, as you can see. There are beams that flex. Okay, civil engineers, aerospace, mechanical engineers can understand how beams deform. These are non-linear, meaning that they are undergoing large displacement and large rotation. But we can design them and try to make them at micro scale because there are no joints using lithography. That's where we have our electronic microfabrication, right? So those are the methods that people use in both microfluidics and microelectromechanical systems in general. So those are the tools that we use to make these devices. Okay, and with these we can actually perform all these manipulations, grasping, squeezing, just piercing or injecting. We can roll, we can peel, we can bend, probe, and also stretch. We can hold this and then stretch like this also. Okay, there are lots of manipulations we can do, and I'll tell you why we need to do manipulations. You probably understood why manipulation is needed from these analogies of buying a vegetable or a doctor doing palpitation. Okay, so here is the grasping. Okay, I hope the video plays. Okay, this is just a glass pipette to position the cell. Now we are zooming in, and you can see that we are gently grasping a breast cancer cell, which will be about it's a MCF7 cell line, this one, and it's about 10 microns, something like that. So there's of course cells vary in size, but of that order, we can actually grasp it. And this is SU8 polymer, and when we do it, we're not damaging the cell. That is the thing. We have the stiffness of the device, not only the size, but also the stiffness should match that of the cell. So, for example, if you are holding something very delicate with your hand should have the sensors so that you don't apply too much force to crush it. Okay, similar thing we have to do here. And we can also squeeze. And this is zebrafish embryo. It's a cell, nevertheless, but it is not as like the previous cell. It's quite big. Maybe 100 microns or so. So you can see, I'll probably play this video again. So you would see that we are squeezing and it's recovering real time. All right, so cells are very resilient because they have to anyway go through capillaries and they have to flex and come back. If you imagine red blood cells, right? So they have to go to small capillaries, which are much smaller than their usual size. So they have to squeeze through and then they recover. They're very resilient that way. We can pierce, of course. So we are piercing. This is also zebrafish embryo. We are piercing with a micro needle, and we are gripping with a gripper. In both cases, we can measure these forces. Okay? Measure this piercing thing as well as this. For example, when you do intracytoplasmic sperm injection into a cell for fertilization, this is what we call in vitro fertilization. No glass now, but still called people it in vitro fertilization. So when you're injecting a sperm into an egg cell, you have to go into, let's say, nucleus, right? So you have to feel that. You have cell membrane will be softer than a nucleus. When you can see it, but then that's not scalable. So if you have a tool that measures the force, that will be useful. Okay. Okay. So let me. Okay, I think this has stopped. Hello. Yeah, it's not going to the next slide. Okay. Yeah. So we can also do this rolling. Why do you need rolling? I already explained. We have to look at this stiffness in multiple directions. You can see here in this video how to roll a cell. You are holding with your like 2 fingers, and then you are doing something like this to roll. Okay. And that can also be done. Okay. And we can peel. These are like chondrocytes, corneocytes, so which we are trying to use it for a cosmetic company, and that's where we're doing. That's not so relevant to the talk. I'll just skip those. We can actually peel a cell, measure the peeling force. We can bend a cell and look at its stiffness and so forth. Okay, and we can probe, of course, that is easy. We can also stretch. So for stretching, it is a little bit more difficult because a cell is sitting suspended. It's trypsinized so that it's not adhering to a surface. We can manipulate both cells at the single cell level, things that are adhering to a surface as well as things that are floating. The stretching is a little more difficult because you have to grab it and then stretch it. So here we have to do two levels of things. There is a major one, like we sell a compliant mechanism that's of millimeter size, and there is a micron size thing inside. And two things made differently, assembled together, we're able to sketch. And this video that I'm showing might will make it clear for you how we can stretch a cell. This is a big one, so you see, just to show the concept. I'm pushing here, and these things touch, and these things open up. So now, next part of the video, we're going to place a piece of rubber band here. We are pressing above somewhere. The moment they touch, these things open. When they open, you put a rubber band or something. And then after that, if you leave it, it's going to come. And then when you press again, it's going to stretch. As you will see, this piece of rubber band will be added here. And now when I press it again, you can see the rubber band being stretched. Okay, we can stretch a single cell. Of course, now we are doing differently. This is just early work. We are doing single cell, but now we do in a scalable fashion also. This is the compliant mechanism that with which you can actually roll. So you can see by having two different inputs to these points, we can actually grasp something as well as roll it. Okay, and these are our setup for doing all this. So we have uh, basically inverted microscope and controllers to move these, and then things are over here. Okay. And how flexible is a biological cell? That is something that we need to first understand. How flexible is a biological cell? When I say flexible or stiff, I need to have a unit to measure. It is Newton per meter. That is, if I apply certain amount of force, how much displacement do I cause to the object? Again, we go back to the tomato example. When I apply some force with your hand, maybe you can apply a few newtons, and tomato may, deflect by maybe 100 microns or a millimeter. Okay, so based on that, you will know what is the stiffness of the tomato. Okay, in a similar way, if you think about a ballpoint pen, okay, there is a spring in that. If you say, what is the stiffness of that ballpoint pen spring? So let's assume that you can compress it by 1 centimeter by applying some force. Let's say 1 Newton. It will be a little bit more than a Newton, but let's say 1 Newton. 1 Newton force is needed to deflect it by a centimeter, or 100 Newtons for a meter, Newton per meter if we take. If this is the stiffness of the spring. Now, what is stiffness of biological cell? It turns out that stiffness of biological cell is around millinewton per meter, 0.001 Newton per meter. Compared to ballpoint pen spring, will be 1 lakh times less stiff. Okay, this 100, this is 1 by 1,000. Okay, cells are one of the softest things a mechanical engineer can do. deal with, very flexible ones. Okay. So now, how do you measure such small stiffness? We have these tools now with which you can stretch or compress and find this. We do that measuring in a similar way that we weigh vegetables, right? You go to the spring balance, put something there, the spring deflects, the spring stiffness, spring constant as we call it, and multiply by displacement, that is spring stretching, and based on that we say this is how much force is applied on the spring. In a similar way, we observe the displacement of a few points, okay, such as those here, and infer from it what the force is. Okay, so our device is, big one is 10 millimeter, 10 millimeters. The small one that goes here is 1.7 by 1.7. And these beams are only 5 microns in width here. Okay. And there's a process, we use lithography based process to make these things. Okay. And with these things, we can measure this S8 micro gripper is the one that enables us to measure the thickness of cells. Okay. And if you think that, oh, it's all new, no, okay, we go back to 1939 paper by somebody called Charles Hamilton Norris. So where he had measured the tension of nucleated erythrocyte. Normally, red blood cells, erythrocytes, do not have nucleus, but at the beginning when they're made, they will have a nucleus. They lose it when they squeeze through some capillaries before they enter the bloodstream. Okay, they took the nucleated erythrocyte and measured the stiffness. Amazingly, the value that they give is 1.4 millinewton per meter. Of course, their paper gives in dynes and centimeters. We convert it to our standard units now. 1.4 milliliter per meter. That's exactly right. So they had done it in 1939. So they had this cell here and a pivoted glass needle and liver. So when they move it like this, they had a way of measuring the force and also the displacement magnified, and they were able to get the stiffness. Very ingenious at the time. Now we just have done it, with modern tools that we have. So our tools are, as I said, compliant mechanisms, and with which we can do this. Okay, so here is where you see this was actually a zebrafish embryo, and whose stiffness we determined 8 Newton per meter. Of course, it's 1,000 times more than single cells that we would deal with, but this is embryo that has several things. Okay, this is how we try to measure force like a spring balance method. Okay, and okay, once we do that, we worry about the specificity. Okay, let's say I take a cell and I decide that it feels stiffer than normal. So what can we make out of that? So here we did this problem of inverse problem, as we call it, where we try to measure displacement at a few points, try to infer the stiffness. This inverse problem is more in terms of way of determining the stiffness from displacement that is measured, more than a spring balance type of work to measure force. A little bit more elaborate. I don't want to get into details of that. Okay. The second one that we want to do that, another inverse problem that we have is actually, this is just to measure the force. So let me just finish that. We have made this. There's a computational thing behind. It's a vision-based sensor. Basically, we look at image of the cell and our tool. By measuring the displacements of certain points on our tool, we can measure displacements and then use our computation to understand the stiffness. Okay, that's the first part. There is a entire device also to that extent. Now it is kind of called micron, MICOR capital N, just a micronewton force sensor. Okay, this is actually now like a packaged device with which we can do this. Okay. Um, but really, there's a second inverse problem that I talk about in terms of the specificity. That is, if the stiffness has changed, what caused the stiffness change? Is the cell membrane has become stiffer, or the cell nuclear envelope has become stiffer, or nucleus has become stiffer, or cytoplasm has become stiffer? There are, I mean, at least I'm broad categorization I'm doing, but lots of things that can happen. So we want to do this elastic mapping inside the cell. Okay, so what has changed? That's what I'll come to. Okay, let me just start with a particular case study. this WebEx, I'm not familiar how to get rid of it. It goes by itself at some point. Otherwise, it's blocking. Yeah. So usually whenever you do, a multimodal approach is good. So normally mechanical manipulation, if you think about our characterization, you want to do compression, tension, and shear, the three modes that are important. Okay, that's what we are doing for MCF7 cell line. These are breast cancer cells. And how do you do compression? How do you tension? How do you do shear? Okay, so here we have introduced some biochemical stimuli. Okay, the control cells meaning they are the normal cells that we have taken. Breast cancers, of course, they are breast cancer cell line, but as they are. Okay, the blue refers to the nucleus of a cell, and the red is actin, which is cortical actin, which is at the cell membrane. You can see here, it's only the dye for blue here, nucleus. This is all the cortical actin. And then the green here is microtubules. All of them put together, it will be like this. There's nucleus, microtubules, and then actin filaments. Okay, actin filaments decide the stiffness of the cell membrane because the cortical actin is outside of the uh, cell and membrane. Microtubules are the cytoskeletal, both actin and microtubule are skeleton of the cell, cytoskeleton. So they will be there. They'll be there between nucleus and the cell membrane. But there are lots of other things that are the main ones. Now, when you subject the cells to this cytokeratin D and nocodazole, both are cancer drugs basically. Okay, they disrupt the cell processes. The cytokeratin D, as you can see, it disrupts actin. Microtubule is intact, nucleus is mostly intact, but the cytokeratin D has disrupted this actin. The cell membrane becomes very, very flexible. It's all gone, right? As you can see. So that's one biochemical cue that we do. The second one is nocodazole. Here, what happens? Nucleus is fine, and this is not the same cell, by the way. That's why they don't look the same. Okay, this actin is fine, but microtubule has been disrupted. Okay, so there is no connection between nucleus and no, I will not say no connection, but there's something disrupted with the connection between nucleus and the cortical actin of the cell. Okay, so we have these things and the control to compare what happens. So for the tension, we have our own tools, but then instead of using our tools because they need to be calibrated and established before we collect data, so we use the standard techniques, which is micropipette aspiration. So we have these micro tubes, small glass needles, pipette, right? It's a small pipe, and the cell is sucked into it by applying a suction pressure. So here we have control, normal things that the MCF7 cells, and then cytokeratin D, which disrupts actin, and then nocodazole disrupts microtubule. After a while, that effect goes away. So we have done nocodazole, and then that effect goes away after some time. So we try to see the aspiration force. That is suction pressure multiplied by the area of cross section gives us the aspiration force, which is nanonewton. Very tiny force you need to do to suck a cell. The cell is so flexible, as you saw the number, millinewton per meter. So control if it has so much, less is enough because cytokeratin D and nocodazole both change the mechanical stiffness of the cell. Okay, in this case, actin, this is microtubules. After some time, the nocodazole effect goes away. Microtubules are reformed again, and then stiffness or aspiration force required to suck into the tube increases. So there is a good correlation here. Okay, and how do you do compression? You have to press on it like a spring that you press. Here we use AFM, atomic force microscope, which can press on a cell. Okay, cell is now on a substrate. There is nucleus, and the cell really spreads on it. And you have to be careful where you press. That's why you've taken a 4x4 grid. Identify a point right above the nucleus. If you do it here, you'll feel most stiffness of the substrate, not the cell. Okay, we do it over there. Again, control has, there is a way that we make some approximation and say Young's modulus of the whole cell, which is a gross approximation. Okay? But it's a good indicator. It's like a material property of the cell, gross approximation. if the control thing without these chemicals put in, if it is so much, by putting actin, by disrupting actin, it comes down drastically. But nocodazole, it goes up. Why does it go up? Because the moment actin, which is in the cell membrane, goes away, AFM starts feeling the stiffness of the nucleus. Nucleus is stiffer than the cell. Okay, so if this is axial so much, it actually goes up. After nocodazole treatment goes, there's a slight difference here. Okay, but the fact is that we can actually see if the stiffness has increased, okay, of the cell, we can see that either it's cytokeratin D or nocodazole. That's a kind of bare specificity is there. And how do you do shear? Here I'm not talking about fluidic tools, but that's also useful, especially if you are culturing cell in a channel over which you flow the medium. We have made perfusion bioreactors also, especially for liver cells, which I'll be coming to later. So we flow that, and we can see that compared to control, cytokeratin D, when this is there, what we are showing here is deformation index. How much does the cell deform when something flows over it because of shear stress? Okay, eccentricity basically increases here. So you can see that cytokeratin D treated cells have higher deformation. Nocodazole, where the microtubule disrupted, it is even higher. Okay, and of course, when it recovers, it comes down again. Okay, we can actually see with all these things, we can actually see what might have changed. Okay, that's one part of it. Let me go to the computational techniques now. What do you make out of these, right? If I have a cell, okay, some that is normal, another one that is not normal. Somebody tells us, okay, here is a cell that are subject to this virus or bacteria, or whatever, or some condition. Okay, and when we measure the stiffness of these two, if it is identical, for example, that's not good for us, right? So we have a healthy cell and an unhealthy cell, and if both of them have the same stiffness, how do you tell it apart? That's where we actually do this. If they have the same stiffness, and then we ask, what causes the stiffness of the cell? Okay, so here is where we try to ask this question about specificity. What changed the bulk stiffness that we're measuring? Okay, if these two have the same stiffness, how do you tell it apart? We here do another inverse problem, which is called Dirichlet to Neumann map. All it says is that from the boundary measurements, can you make out what might have caused inside? Okay, so which means that if I were to have this just two these that we have made, for our experiments to understand the problem. So one is this clear material is basically very flexible. In that, we included a hard inclusion. Okay. Now imagine a 2D version of the problem. You're only allowed to touch on the periphery. Okay. You can feel it whichever way you want. I give you two discs like this. They're not balls, they're discs. Okay. I give you can, press on the periphery only. You have to tell which one has inclusion, which does not have inclusion. It is easy, right? Because if you apply some force, the one with inclusion would deform less than the other one. Okay. But then if you have this type of thing, uniqueness is very important. You have to do the elastic mapping. That's a detail. I'll skip it today. Okay, so I'll skip these details. Let's say I have one inclusion. I apply force in various ways. How to get this back? Okay, by looking at the force and displacement only on the boundary, I should be able to say how elasticity, so to speak, is distributed inside the cell. We can't see. It's more like non-destructive testing of cells. There are two inclusions. If I were to say where they are, what shape, and what is the stiffness of those inclusions. And that's what we have been doing. So if we put an inclusion of that shape, we actually get that back. Okay, by looking at displacement and force only on the periphery. And that's what we do. And this now we try to apply to a particular example. Now we go to this biomechanics of liver cells that are affected by hepatitis C virus. Okay, this was the work done by Srinath Balakrishnan and my colleague Prof. Mitra Das in microbiology and cell biology in IISc. Okay, so here we took a model system, which is this human hepatocytes, HUH7 cells. Okay, that's in gray color. Remember this color because other slides would have this color. And then these orange ones, which are harboring what are called hepatitis C virus replicant cells. Okay, these are the ones which, if you look at the HCV RNA, okay, so replicant cells would not have the structural proteins in them. Rest of them will be there. They can replicate and do all that. They are not really infectious like the original ones are. Let's call them replicant cells, which have hepatitis C virus, but in a way that it is not infectious. Okay, you can just characterize some things are taken out of that. All right. So we have, this is the control, and this is our study thing. Okay. So here, what we noticed was that first the nuclear morphology. So look at the cell, but then cell has nucleus inside, cytoskeleton, lots of things. You have to focus on something. Here we decided to focus on the nucleus of HCV replicant cells as well as normal hepatocytes. Okay, so if you look at that, well, then we see this is the top view, this is side view, and then this is another side view. What we have, mechanical engineering have these three views of a machine, right? Top view, side view, and elevation. Okay? Similarly, we took this confocal microscope gives you this. Okay, if you look at this again, nucleus here is put in green, not like blue in the earlier slides. Actin is still red. Okay, these are the two dyes. If we see one cell, of course, we looked at hundreds of cells. We notice that in HCV replicon, nuclei are larger. Not only the top view or projected view, look at the microscope, okay, but also confocal microscope gives you the 3D view. Okay, it looks thicker than this. I'm just showing one image, a representative image of the two. But looked at lots of cells. Nucleus here seems to be bigger, both in the area in the projected view as well as the height. In other words, volume has increased. So nuclei in HCV replicon cells have become bigger than the other one. It is not even stiffness. Just look at the morphology first. Just the geometry. Normally, biologists will look at it and then they may infer something, may not infer something. But for a mechanical engineer, that seems significant for us to probe. So we actually made a bioreactor and peristaltic pump, everything ourselves. So we can do a perfusion reactor so that we imitate the condition of a liver cell. I said when engineers work on biology problems, we make our own tools as well because that's what we used to. We made this. Now it's actually available for others to use as well. There's a laboratory setup, but now we have a commercial version that BenFlex gives to people, so we can do perfusion in various ways that we want to do. Okay, this is the commercialized version. Okay, now if you look at the cell population that we have, we take the nuclei of these and try to determine their projected view as well as volume. Okay, we get the edge of the cell here, and as you can see, this is an image processing algorithm that holds onto that and gets the perimeter. Similarly, we can do surface. Okay, so once you know the surface, we know Gauss divergence theorem to get the volume. So if you take this image, a three-dimensional confocal image processing runs on it. This is the surface area and the volume. Okay, so we try to plot these things: the projected area, the volume, and the eccentricity of the nucleus. Okay, of course, idealized. It's not an ellipse, but ellipsoid, but some arbitrary shape. But we can approximately get that eccentricity. That is this 2a by 2b, or a by b. Our eccentricity, we actually measure this square root of 1 minus b square by a square. Okay. Now, how many nuclei of, again, gray color is the control, and orange is the HCV replica. What you notice here, HCV proteins increase area and volume and decrease eccentricity of the nucleus. Okay, here as you can see, project area has increased from this mean to this mean, and the volume has increased from here to here. So project area has increased, volume that we had seen with the eye, now we have data. Okay, lots and lots of cells, hundreds of cells, thousands of cells. Ugly 461 here and 327 there. Okay, and eccentricity is other way around. It has come back this way. That is, the normal HUS7 cells have higher eccentricity than the replicant cells. Okay, what do you make of that? Okay, things have become bigger, but they're more spherical now. They're not, like ellipsoids. Okay, meaning that if something like a sphere ball is there, okay, if there is no force or reduced force on it, it won't become like an ellipsoid. That told us something is happening with the force somewhere. Their nuclei of HCV replicant cells are bigger and they are experiencing less force. That's why they are not ellipsoidal, they're more spherical. Eccentricity has reduced. That prompted us to look at this model. Okay, think of the nucleus as a ball, and then there is force coming. The force is coming from the actin. Actin is the one that pushes down on the nucleus. Okay, that force might have reduced, or the nuclear envelope might have become more flexible. That's why the volume has increased and its eccentricity has decreased. That actually prompted us to look at the modeling first. Okay, what happens when you apply the force? Instead of becoming this way, it is less, so there is less force. Okay, and of course, we estimated the nuclear envelope modulus also with these numbers. Okay, from 26 came down to 13, from HUH7 to HCV replicant cells. Okay, and we also did AFM, atomic force microscope, and found that this force, okay, where we measured force versus displacement, what we found was compared to control cells, these replicant cells, apparent model of elasticity increased. Okay, so on the one hand, nuclear envelope, nucleus is increasing in size, becoming less elliptical, and we find that the cell stiffness has gone up. Okay, on the other hand, if we were to put this microtubules disruption, okay, then again replicant cells have nuclei have less stiffness. Okay, when you take out the microtubules, that tells us that okay, microtubule also has a role to play. So there are conflicting cues that we have. We need to put the piece of the puzzle together. So what we did was, okay, one thing that we see is that cell membrane is becoming stiffer. Okay, and the nucleus is becoming larger. What could have happened? We try to see molecular basis. Normally, biologists, whenever they find something like this, they try to probe which protein. They do protein array analysis. We see which protein. Look at lots and lots of proteins and say, oh, this is upregulated, this is downregulated, and so forth. Here, we didn't have to do that. In fact, this was enough for us to suspect that uh, there are these lamin AC proteins. Okay, nucleus has become bigger. Nucleus has become bigger. How can it become bigger? There is, of course, osmotic pressure and all this pressure that inflates the nucleus. Okay, and pressures more or less the same. It has become bigger. That envelope flexibility might have increased. And what controls the stiffness of the nuclear envelope for these lamin AC proteins? We directly said, okay, let's look at lamin AC. We didn't do all the protein array. Just look at lamin AC, and they were downregulated. Okay, whereas this beta actin that we got, they were upregulated because with the cell membrane stiffness has gone up that we have found with AFM, mechanical response. Okay, so we could pinpoint this laminacy in beta actin in much shorter. So biology research also can benefit if you include mechanical response, is what we found out from this study. And we did this for other cells as well as other viruses also. It's not HCV, but others. We need to look at in biology because people are very suspicious because there are a lot of variability. We did all those negative positive controls and everything, and then established that lamin AC is downregulated, beta actin is upregulated. Okay, and that is the thing we found. All right, and we also did a shear flow test. So all these cells are here, we are flowing medium over them, and we find that the extent of the nuclei of a replicant cell increases under physiological shear stress. The flow rate was such that it applies physiologically meaningful shear stress like we find in liver cells. Okay, inside liver there's perfusion. We get the same thing in our flow experiment. Here we see that replicant cell stiffness uh, increases. Eccentricity of the nuclei of cell increases, meaning they have become more flexible. The nuclear envelope has become more flexible, which is what we suspected. Okay, with all these things now, we try to understand what might. And then another thing that we learned from our biology colleague, Professor Pamitra Das, was that in HCV, normally what they have found, if hepatitis C virus affects liver cells, some proteins which are not inside the nucleus are found there, and which are not supposed to be in the cytoplasm are found there outside the nucleus, which means that the nuclear envelope has become more flexible, is what we find mechanically, but it also become more permeable to proteins. Okay, so it could be, we have not established it firmly, it could be that the virus is making the nuclear envelope more permeable, meaning things can go back and forth to do its job. Okay, such insights also come out from the mechanical studies. Okay, now heterogeneity. So just to close one the talk now, um, biologists always say cells are very heterogeneous. There are lots of things, and so are machines. So are many materials. They're not the same, but we have a grasp because we try to look for general underlying principles. Okay, so with that view, we try to see, okay, if we look at nuclei of HUH7, there are lots of different sizes and shapes. There is heterogeneity. Okay, but there is anything common to these things? Can we say anything about whether they all belong to the same type of state of health or not? Okay, so Srinath did this experiment. Of course, this I have shown. Projected area, surface area, and volume all have increased from the control cells of HUH7 to the HCV affected hepatocytes. Okay, and we have mechanical model, very simple model. Things have to be simple in order to be effective. So we took a model. This is actually a model that was taken from tires, no? The vehicle tires, because they're also inflated. Okay, they are inflated, right? And there is a force that comes on them, the vehicle's load. So the cell is also similar. There is pressure that makes the nucleus kind of inflated. At the same time, the actin, like a stretched rubber sheet, pushes down on it. So simplified model, a cell which is spherical with the pressure that is keeping it inflated, and then the actin, this membrane, pushes down like a plate. We took this model and tried to define two non-dimensional parameters. Okay, P is the pressure, R is the radius. Okay, you only see nucleus like this, but if you were to let it be free, it become a ball or a sphere. The surface tension, that is the radius R. E1 is the nuclear envelope stiffness captured with just one number, which is a gross approximation. And this P, of course, is the pressure, and then force is the one that is coming over here. Okay? The force that presses it down, which is relative of indicative of the actin, cortical actin force that's coming. Okay? So now what we find is that if I were to take lots and lots of cells, again the gray indicates the control, orange indicates HCV replicant cells, put this in a three-dimensional plot. There's a surface area normalized, projected area normalized, and volume also normalized. Okay. When you do that, we have a surface that the model gave us. All the cells lie on that surface. Doesn't matter nuclei. I mean, it doesn't matter what shape they have, what size they have. They all lined up. And you can see that we have this everything going along this direction. Now we have further work on it. It's a paper under review. We have identified two parameters, one that goes along this direction, other goes along that direction. Now you see normal things have moved here, something has changed in the cell. Okay, quantification we can do. All right. And if it actually goes along this line, now we have done a lot of other cells, HeLa cells, fibroblasts, and a lot of other cells. They all occupy different lines on this surface. Okay, that was exciting for us. Where we also done AFM measurement to show all these results. What has become stiff, less stiff, or whatever. Okay. Now, by looking at these nuclei of these cells, we can tell what category of things they belong to. And the same cell, when it moves along this, so they line up. Okay, that is one thing. Types of nuclei. We can say this belongs to that cell, this cell, this cell, and then something here moves along this. There is something has changed in them. Move that way, something has changed in them. So now we're able to do mechanodiagnostics simply by looking at the nuclear morphology. Okay, so just the last portion, which is engineering tools for interventions to do on the cells. I showed these things earlier experiments where you take a single cell and then squeeze, stretch, and do all these things. Now we want to do in situ scaling. Because these days everything people want to scale. You cannot do one and then say, yeah, I've done it. It should be scalable. So we have done this now with a cell stretcher. This is a compliant mechanism. You can see one side it compresses, other side it stretches. Okay, so which is what we have done. We have put these uh, what we call cell stretchers and cultured cells on them. You see nuclei. Okay, the cells are here. I'll play a video where you can see we thought that when we put these cells over there, we can stretch it like we are showing here with a probe. We thought we'll stretch the cells and then see how they behave. So a surprise when we cultured cells on these, our device was so flexible that cells could apply force to stretch and contract. Okay, as you can see here in this video, the cell is here that actually detaches and then divides and then attaches again. So the force they apply decreases and then increases when the cell divides and grows. We're able to do that. It's a real-time video. Of course, this is see 600, 700 minutes, like 2 hours. Okay, where we were able to see this in situ stretching of the cells. And this just to show that the force decreases and then increases when the cell kind of stretches it and then detaches when the force comes down and then divides again and attaches when the force goes up again. So we're able to do these things now. Now we're also doing other thing using what people call now micro robots or rafts basically. We can make with SU8 polymer shapes and sizes, various things of these things. And what you see here, these particles are magnetic nanoparticles. By applying magnetic field, we can move these around. That's why they're called robots. And what we can do is grow cells on top of them. So normally people are used to doing culturing of cells of one kind. Now we can do one kind and one, another kind, another one, bring them together, and then see how the cells will interact. The cell-to-cell junctions form, what else they do, and that's what we are doing. Here we can assemble cells. This is just a micro robot. This video shows that they can be moved. They're all moving together. They all rotate. They all go this way. They all go that way. But then we have local magnetic fields with separate coils where we can move them the way we want. So this is turning. This is turning. If I activate these coils, if I don't activate this, it doesn't. Now this moves. That stays stationary. So we can actually make these tiny coils. We are using this X-ray synchrotron that is there in RR-CAT in Indore. Dr. Vishal Tamgai has made it for us. We can make tiny coils and create local magnetic fields. We can culture cells on them. I showed the force measuring in one direction. Now we can measure in 3 dimensions also by making these little flexible spirals where we have cultured. Again, nucleus is blue, actin is red, and we can culture lots and lots of them. Okay. Now we can also make these breathing scaffolds. I said we try to create in vivo conditions. You see here, one of them, this one is stretching in plane. Okay, we'll see that. It's like contracting and expanding here. This is going out of plane, like gut on a cell, uh, uh, chip, gut on a chip, lung on a chip. People talk about, we're able to do that. We can make these breathing scaffolds for these cells. And so I've described the tools and the techniques. And last one is there, discrete element method, and 2 case studies that you do. The main points is that mechanical response can be measured. Okay, that is something we have established, and so are many others elsewhere. Okay, and we want to go with specificity. It's not like we ignore it. We want to do diagnostic specificity. I showed a couple of examples to that effect. We want to understand biological mechanisms also. Like in the liver cell study, we're able to get the proteins, rather easily because we know how this particular nuclei is behaving. So lamin AC and cortical beta actin were pinpointed by mechanical characterization studies rather than doing protein array analysis. Okay, and we can mechanically intervene, like what I'm just showing. That's ongoing work, micro robots, and some of these tests that we are trying to do. Okay, And biological processes also can be discerned using mechanical studies. A lot of students, I only showed some names, a lot of funding agencies, all this contributed to this particular work. And finally, I would like to talk about this, just 2 slides. I started out saying cells are very granular. There are lots of things inside of all kinds of shapes and sizes, like Crick's analogy. They're all moving inside. Now we're making these tools using the discrete element method. There are these different particles, and we can try to see, like the study that I said, if I were to have a cell membrane, nuclear envelope, nucleoplasm, cytoplasm, different colors here. When I do it, how does it move? Now PhD Prasenjit is kind of doing it. And some of these things are possible with the discrete element method. For example, this study of a mitochondria which undergoes fission, meaning something that is called DNA protein, which is like a helical thing. It actually squeezes this mitochondria like boa constrictor, breaks into pieces. Okay, so this is something that we are trying to do with discrete element method. So things happen under certain disease conditions. I think Alzheimer's also one cause where people have found that mitochondria exists in little pieces rather than together. Maybe mitochondria wants to uh, have more exposed area to do the job better. Okay, it breaks into pieces, and what breaks into pieces is this DNA protein that actually squeezes it, breaks into pieces. It's a very mechanical process. When I see such things, then I see that mechanical engineers have a role to play in the sense that life is very mechanical. Okay. So let me stop here and take any questions. I hope all the videos played in real time for you. Yeah, thank you.
Uh,
I'm done. Yeah.
Yes, sir. Thank you, thank you, sir. Thank you so much for such a wonderful talk. It's always a pleasure to hear you. So, any questions from the attendees? Sir, we have students from our BTech Biotechnology, Mechanical, and Electrical Engineering students. Also, we have faculties from
Sure.
other institutes all over India.
Yeah.
So, yeah. So, attendees, this is the right time to shoot your questions to Professor Ananthasuresh. He'll be more than happy to answer your queries.
Any questions, comments? What do you think? Would mechanodiagnostics be ever possible?
Sir, actually, I have a question regarding this mechanodiagnostics. You were talking about the sorting the cell depending upon the physical properties.
Mhm.
The same thing we have been doing for the FACS, which is a fluorescent activator cell sorter.
Mhm.
So how different, of course, you are not using a fluorescent dyes in sorting the cell.
Mhm.
So how different is your mechanodiagnostic one? How efficient it is compared to the FACS?
Yeah, fax is more like an imaging technique, right? That just, you have to put the dye, you have to have intervention. Here it's uh, basically mechanicals. In fact, in the scalable thing, we want to do in a micro channel when cells are flowing, for example, trypsinized cells. By just looking at each one of them, we can do thousands of cells at the same time. I had another PhD student with a colleague, Prasenjit Sen. We are trying to do that also, okay, with a squeeze.
Okay.
And now with the little grippers also, we can do multiple things. I showed this cell structure device, culture all the cells on top of this scaffold. Now our scaffold itself has sensing capability. Okay,
Okay.
so
Oh,
other cells are culturing, we can see what's happening to them, what forces they're applying, and what forces we can apply on them. The micro robots, the magnetic nanoparticles, that's what we're trying to do. We can do, you just take a cells and put on our scaffold, we know what they are. We don't need to put anything else. So it's actually will be eventually faster than what fax can do, and less
oh,
expensive also.
it's
Yeah,
yeah.
but we are not doing the
Thank you for
same
that.
thing. Yeah. Here we tend
Okay.
to do mechanical response, whereas fax cannot do mechanical response, right?
Yes, sir. Thank you. Thank you for the clarification, sir. Any questions? Any comments? Okay. Sir, some students were inquiring, they are from this BTech background, BTech in biotechnology. They are inquiring there if they want to pursue a degree actually in something interdisciplinary one, the one you are currently doing. What exactly they have to, like, subjects they have to focus on? Like, can you comment on them?
Yeah, so we have this biosystem science and engineering department in IISc where we take students from many different backgrounds, from pharmacy and microbiology and chemistry, physics, engineering, all disciplines. And not only this particular department, also other departments also in IISc. Okay, a lot of people work on biology. Biology is very hard today because there are a lot of unsolved problems people think, and everybody has a way of looking at biology in their own way. Okay, what do you need to have? Actually, nothing, your interest. Okay, whatever your skills are, there is somebody in IASC who can use your skill. We have math biology program. If you are interested in mathematical aspects of biology, we have our math department runs that math biology program. So that also is there. Whether you're interested in maths, physics, chemistry, of course, biology, and any engineering discipline, we have people in civil engineering and aerospace engineering interested in these things. So all backgrounds are relevant for this type of work today. Okay, so if somebody, especially in biosystem science and engineering, what we have is a system where we have two advisors, one from engineering, other from biology. Our core faculty in that, they're already bioengineers, so there is only one advisor. So if you are a conventional biologist or a conventional engineering student, then biosystems engineering may be a good program for you because there'll be two advisors. One advisor will take care of your original field, other advisor will teach you the other thing. Okay, so basically just about anybody can do this now. Your background is suitable for a particular study, and if you look at IISc website for these departments, you would know the kind of backgrounds of the students who are taken. All right, so you just
Yes, sir.
write to the faculty, they will tell you where to apply. As a student to IISc, you can apply to 3 departments. Okay, so we have actually not, I will not say 3 departments, 3 programs. We have more programs than departments because of interdisciplinary
Yes,
programs are there. Okay, so we have 3. So if you are a chemical engineer, you can apply to chemical engineering and apply to 2 others. Okay.
yes, sir. Yeah. So there's another question from the students. It's a general question actually. So they're asking, are you interested in taking interns actually for
Yeah, normally we all take a lot of interns, but this pandemic has kind of put a break on that because we ourselves are not operating our labs to full capacity even now. Okay, only 50% capacity. Students are taking shifts to work. Okay, uh, so that's why I'm not saying that we can take now, because there are also other, maybe you're vaccinated, otherwise we're still not completely safe, right? After this is over, yes, you are welcome. We have lots and lots of interns. In fact, some departments have specialized summer internship programs, like Biosystem Science and Engineering has a program called BEST, Bioengineering Summer Training, BEST, and
yes,
likewise mechanical has summer program and other things. Okay, there are lots of possibilities, but not right now.
yes, sir. Thanks for the clarification, sir. Any questions coming from the attendees?
Sir, I have just a small query. I'm not from the biological background or mechanical background, sir. So, like yesterday, I had a talk of, I think, Dr. Shubhashish Halda. So he was using some this thing, optical tweezers and the magnetic
Covalent tweezers, yeah.
Yeah. So, your group is also using such kind of characterization tools, like in the sense, like he was doing multiple characterizations from that optical tweezer actually. Not only mechanical, he was able to do thermal also, and I think
Yeah,
electrical also. So,
yeah, right. I mean, optical tweezers can be
yeah,
used as well. Okay,
yeah,
but our approach has been that anything that you use an external energy, right? It's bound to change the cell. Already you are taking cell from its native environment and separating out and studying it. So when you use optical tweezers, you tend to heat them up a little bit.
yeah,
Okay, that also be taken out, right? So our mechanical approach is minimally invasive in some sense. So people do with electrical energy, magnetic things. Magnetic is also fine. That's why most of the micro robotics work applied to cells is using magnetic field, because among the different forces, magnetic forces are apparently least intrusive on the cell's innate processes. Okay, so mechanical and magnetic forces is what we are using right now. Thermal, electrical, any other things are going to interfere. So we don't want to do that. because you might infer other things. So, but optical tweezers is an established technique, but then I'm yet to see where it is used for diagnostics. It will take time. So all
Yes.
techniques are to be pursued by everybody, but our approach is not to use any other energy other than just purely mechanical.
Okay, sir. Thank you, sir.
Thank you, sir. So, any other questions coming from the attendees? Actually, this is the right time to ask to Professor Ananthasuresh. He is in middle of a lot of meetings, and he is a little bit busy schedule. He is taking time for us. So please shoot up your questions. Okay.
Okay.
So if there are no other questions coming up, then I will take this opportunity to thank our esteemed speaker, Professor Ananthasuresh, for sparing his valuable time and sharing his wonderful research topics with us. Definitely it pays, actually, from his talk, I can say that it really pays to have a kind of a different vantage point, because as a biologist, I'll be looking at the cells in a different angle, and sir is a mechanical engineer. He is definitely looking at the cell in a different angle, actually. So it really pays to have a kind of interdisciplinary collaborations, and I think that is the way forward. Sir, thank you once again for your wonderful lecture. It was a really pleasure to have you with us. Thank you once again, sir.
Thank you.
Thank you
all.
very much,
Thank you,
sir.
sir.
Thank you very much.
Thank you, sir.
So participants, Tavish, I think